when cells start to differentiate into these two different cell types. One is called trophectoderm cells. That's the outer layer of the embryo. Those cells are destined to become the placenta. And then you have this kind of ball of cells, cluster of cells on the inside called the inner cell mass. And those will eventually differentiate into the different, all the different cell types of the fetus. And then inside you have this kind of fluid-filled cavity called the blastocele cavity. Okay, so only half of embryos make it to the blastocyst stage. The blastocyst stage is the stage that pre-implantation genetic testing is typically done on in vitro fertilized embryos, where they take a biopsy of about 5 trophectoderm cells and do sequencing or some other genomic assay. That means that there's already been an opportunity for substantial selection before that stage, right? And actually, it's not even all blastocysts. They only, at least in the clinic, they only test blastocysts that have kind of normal, good morphology that they think would be the best candidates for leading to a healthy pregnancy. So to really learn about what is driving, what's causing this arrest of about half of embryos before the blastocyst stage, we have to test embryos at the cleavage stage, including embryos that stop dividing. And so I was recently part of a collaboration to try to do exactly that. This is a collaboration with clinicians at the London Women's Clinic, where they applied sequencing-based pre-implantation genetic testing to embryos regardless of whether they survived or arrested. So they, in addition to the embryo biopsies at the blastocyst stage for the surviving embryos, they also tested entire cleavage stage embryos that stopped dividing. And importantly, for the blastocyst stage embryos, they didn't restrict the analysis only to blastocysts with good morphology. They applied genetic testing kind of across the board. From a practical standpoint, I guess it's impossible to literally test every embryo just based on the staff or whatever that they have. So only a subset of embryos were tested, but importantly, we know which embryos were tested, and it includes embryos that both arrested and did not arrest. So we're getting a sample of all different types of embryos. In addition to the genetic testing, they also performed time-lapse imaging of the embryos. So they're growing these embryos in a dish and recording these time-lapse videos. And so we can then correlate kind of the morphological patterns and cell division patterns that we're observing with the genetic testing results to see if there are any relationships. So time-lapse imaging, I'll get into this a little bit later, but time-lapse imaging is yet another test that is sometimes applied in the context of prenatal pre-implantation testing, and it's debatable whether it actually helps for improving pregnancy outcomes. Okay, so when you do this genetic testing, I showed last class that you get sequencing reads coming from the embryo biopsy. You're aligning that back to a reference genome, and you have some expectation that, okay, if this embryo is chromosomally normal, normalizing for things like the length of the chromosome, GC content, other technical factors, you should see about the same number of reads, the same density of reads, mapping to each of the autosomes, right? And then for the sex chromosomes, depending on the sex of the embryo, you'll see, potentially evidence of XX or XY. If you see too many reads mapping to a chromosome, that can be evidence of chromosome gain or trisomy. And we can actually potentially pull out some additional information. So we focused previous lecture mostly on these meiotic aneuploidies that are originating in the egg. In the case of meiotic aneuploidies, that aneuploidy is transmitted from the egg to the zygote and then transmitted across like all of the subsequent cell divisions. So the entire embryo is going to have that aneuploidy, right? So the biopsy, when you take a biopsy, all of the cells within the biopsy, you would expect to have the same aneuploidy. And so we have some expectation that, okay, you should have, like, in the case of a chromosome gain, 50% more reads mapping to that chromosome, right? You have an extra copy of the chromosome, goes from 2 to 3 copies, and so you should have 50% more reads mapping to that chromosome. If you see kind of that integer level displacement, we interpret that as meiotic origin aneuploidy. Meanwhile, sometimes you'll see situations like this where, okay, here's evidence of chromosome loss on chromosome 13, but it's not like a full, like, reduction, 50% reduction in the number of reads. It's kind of like an intermediate signal. It looks like there are too few reads, but it looks like maybe there's like a mixture of cells in the biopsy. Like you have some chromosomally normal cells with 2 copies of chromosome 3, and then you have some cells that only have 1 copy of chromosome 3. This is a bulk sequencing of about 5 cells. And so when you mix the data together, you get something kind of in between. And so we interpret that as evidence of mosaicism within the biopsy. And if the biopsy is mosaic, then the embryo presumably is mosaic. So we infer that to be a mitotic origin aneuploidy that must have originated sometime during cell division in development of the embryo. That logic makes sense. Okay, so we can, this is a busy slide, but we can separate out the aneuploidies of meiotic origin versus mitotic origin based on whether they show those 2 different signatures. And so on the left, I have the aneuploidies of meiotic origin, on the right of mitotic origin. And this gets to a question that we had last week. Like, are there preferences for particular chromosomes, meiotic aneuploidies or mitotic aneuploidies? Do they tend to occur on particular chromosomes? And we already saw that meiotic aneuploidies seem to kind of preferentially affect chromosomes 15, 16, 21, and 22, whereas mitotic aneuploidies tend to be kind of more evenly distributed across the chromosomes. So we replicate that finding in these data. Here I'm also stratifying by embryos that survived. These are the blastocyst biopsies versus the cleavage stage embryos that stopped dividing. And it's a little bit hard to see in these plots, but I'll show you another figure in a minute. Both meiotic aneuploidies and mitotic aneuploidies occur at a higher rate, or are observed at a higher rate in the arrested embryos compared to the surviving embryos, which implies that there's some like selection happening against aneuploidy even in the pre-implantation stages, even in this first 5 days of development. As we might expect, meiotic aneuploidies have this association with maternal age, but mitotic aneuploidies don't show any significant association with maternal age. But probably the most kind of obvious pattern that we see when we contrast the blastocyst embryo biopsies from the data from the arrested embryos is the number of chromosomes that are affected with an aneuploidy. So when you look at these early stages of development, it's very common to see complex aneuploidies. So embryos with aneuploidies affecting more than 1 chromosome simultaneously. So that's in contrast, for example, to trisomy 21. Individuals with trisomy 21 only have aneuploidy affecting chromosome 21. But if you look in these early embryos, it's very common to see embryos with like, an extra copy of chromosome 21, but also a missing copy of chromosome 4 or whatever. If you look in the arrested embryos, you see an enrichment for these complex forms of aneuploidy with multiple chromosomes simultaneously affected. So here's like an embryo with 10 aneuploid chromosomes. In contrast, for the blastocyst stage embryos, most of those have 0 aneuploid chromosomes. But if, aneuploidy is also common in the blastocyst, but it tends to affect just 1 or 2 or potentially 3 chromosomes. So these like really severe kind of complex aneuploidies are very common in cleavage stage embryos, and they seem to be associated with embryo arrest prior to blastocyst formation. We can fit statistical models to these data because we know like which embryos were tested and from which patients, etc. And so we can fit a logistic regression model and ask, okay, conditional on observing a particular form of aneuploidy, how likely is that embryo to arrest? And what we find there is kind of was surprising to me at first, given the field has focused so much of its attention on these meiotic origin aneuploidies, because those are the ones that are contributing to clinical miscarriage, the ones that are actually observed in clinical setting. But for IVF embryos in pre-implantation stages, it actually seems like these mitotic aneuploidies are playing an outsized role. So embryos with mitotic aneuploidies have a higher rate of arrest. And I think it's a little bit counterintuitive in that you might think, well, okay, meiotic aneuploidies are affecting the whole embryo. So wouldn't that be more harmful than these mitotic aneuploidies that are only affecting a subset of cells? And I think you're right about that, but the thing that is driving this seems to be the complexity of the aneuploidy. So mitotic errors, at least the ones that are observed at this stage, tend to affect multiple chromosomes simultaneously, while meiotic aneuploidies tend to affect just 1 or 2 chromosomes. Questions about that? Okay, so how are these complex mitotic aneuploidies actually originating? Because we have the data from time-lapse, we can correlate the time-lapse patterns with the patterns of aneuploidy and the probability of embryo arrest. What we see is that, and this is not our original finding, but we can show it in more detail here. Embryos, at least in IVF, frequently undergo abnormal initial cell divisions. We don't know if that is an artifact of IVF. However, we do know that, as I mentioned last class, it typically takes even young fertile couples months on average to get pregnant, even when they're trying. And there's also data from pregnancy tests, limited data that suggests that these very early pregnancy losses may be very common. So we don't know how much of this is kind of an artifact of IVF, but at least in IVF, what we see is that it's very common to have, for example, in 10% of embryos, you see these multipolar divisions where rather than the zygote dividing into 2 cells and then into 4 cells, the zygote will directly divide into 3 cells. By disaggregating and doing single cell sequencing on each of these individual cells, what we observe is that the genome actually also segregates on this multipolar mitotic spindle. So you have typically a normal chromosome complement in the zygote, and then each of the daughter cells gets too few chromosomes in kind of a semi-organized way. Obviously, that is going to be lethal when the embryo continues dividing. And so these embryos will eventually arrest. After the first couple of days, about 80% of them have arrested, but these embryos will eventually entirely arrest. Multipolar division is not the only type of abnormal division that we see, though. There are these other really common forms of abnormal division. One of them termed precocious division, where you get normal division from a zygote into two cells, but then a second division that happens too rapidly before the genome would have time to replicate. And so similarly, all of these different forms of abnormal cell division are associated with high rates of complex antigens. So we think that this is kind of the mechanism that is leading to mitotic aneuploidies and causing this really high rate of early pregnancy loss, at least in vitro and potentially in vivo. Yeah, Kurt.
So just in terms of like an evolutionary perspective, how do like embryos learn to arrest? To say like it's better for the survival of like my genes for me to kill myself and another embryo to come along and succeed. How does that like occur in the long term?
Evolutionary explanation. I don't have a great answer for you. Like, I guess one way to think about it would be that these are happening very, very, very early. And so potentially it's effectively like not getting pregnant, right? There hasn't been a huge, maternal investment in these pregnancies yet. And so maybe it would be better for these embryos to arrest early on from like the parent's perspective, if they had no chance of surviving anyway. But like, why are these aneuploidies so common in the first place? I don't have a good explanation for you. In terms of like what's actually mechanistically causing them to arrest, I think it's because embryonic genome activation is occurring around this time, around the 4 to 8 cell stage. And so the first division, second division can happen pretty much regardless of the number of chromosomes. But after that, development kind of increasingly relies on embryonic transcription. And you start getting expression of genes on these chromosomes that are present in abnormal copy number. And that leads to dosage effects where, and it's thought, work in model organisms suggests that the detrimental effects of aneuploidy are mostly caused by stoichiometric imbalance between members of multi-protein complexes that are supposed to be present, like in the right ratios. And if you get abnormal number of like particular components, those proteins can start to aggregate within cells, and those aggregates can be toxic to the cell. And so it's thought that that's like kind of one of the main causes of the deleterious effects of aneuploidy. Good question, though. Okay, so I'm doing this kind of chronologically out of order, but I think it's more logical in terms of the presentation. So I'm backing you up to my PhD, where I worked with the similar type of data in my PhD, but at that time, it was actually common, rather than doing this biopsy at the blastocyst stage of development, taking 5 trophectoderm cells, back prior to the mid-2010s, it was more common to sample from cleavage stage embryos. When the embryo is only 8 cells, they would take a single cell of those embryos and do genetic testing just on a single cell. And hopefully it'll become clear in a minute why that's important, but I used those data to do the same type of analysis where we have the mother's and the father's DNA and to look for genetic variants in the parents' genomes that might influence rates of aneuploidy in the embryos. At that time, it was only about 21,000 embryos and 2,000 sets of patients. So we're scanning through the maternal and paternal genome and looking for genetic variants that are associated with increased or decreased rate of aneuploidy in the embryos. And when we did that, I found my PhD was like finding this one hit on chromosome 4. And you can see that it turns up in the scan of the maternal genome. It's the GWAS on the maternal genotypes and the paternal genotypes. And so when the mother carries a particular allele of this haplotype, she produces a higher rate of aneuploidy in her embryos. When the father carries that allele, it makes no difference. So that's an interesting initial observation. And at first, I said, oh, great, this is like exactly what we're looking for. I came into this looking for those meiotic origin aneuploidies, because there's been so much focus in the field on meiotic aneuploidies occurring in the egg. I was looking for these variants that would turn up in the maternal association study, but not necessarily the paternal genome association study. So it kind of made sense to me in that context. Okay, so what is this hit? Well, first, just to show you the effect size, again, this is GWAS. We tend to find kind of modest effects. This effect is maybe a little bit bigger. It's the equivalent of like a few percent increase in risk of aneuploidy for every copy of the risk allele. So if you have an AA genotype, you have like maybe 10% higher rate of aneuploidy than if you have the GG genotype. You can see here that I've labeled the y-axis mitotic aneuploidy. What we found is that it's actually specifically the mitotic origin aneuploidies that are showing this association. So that was kind of counterintuitive. We found this association in the maternal genome, not the paternal genome, but it's an association with mitotic aneuploidy in the embryos. Anybody have ideas, actually? I'll turn it to you to see if you have ideas. Like, what could explain that? Why? How could we see an association with mitotic aneuploidy that shows up only in the maternal genome versus the paternal genome?
And this was when the testing was still done when the embryo is just that there's no blastocyst, right?
Yeah, these are cleavage stage embryos, so just a single cell.
Does it have to do with the fact that the maternal regulation of the first few divisions versus paternal addition of genome?
That's my interpretation. Yeah, so we think this is a maternal effect allele where, again, these first divisions are not being controlled by the embryo's genome, but by RNAs and proteins deposited into the egg primarily. The sperm actually does make some small contribution of RNAs as well as the centriole, but most of the machinery for these initial mitotic divisions is coming from the egg cytoplasm. Cool. Okay. We also had a little bit of data from blastocyst stage embryos, and actually recently we've replicated the result that I'll show you in a second on a much larger data set. But what we see when we pull in the blastocyst stage embryos is that if you look at this same SNP, patients with the high aneuploidy allele have fewer blastocyst stage embryos that got tested. these are not the same patients, by the way, but look at separate patients. Patients with the aneuploidy risk allele have fewer blastocysts available for testing, which implies to us that whatever this mitotic aneuploidy is, it's causing embryos disproportionately to arrest prior to blastocyst stage, which is consistent with what I showed you on the previous study, right? Again, the effect is, kind of modest, but by around maybe one embryo difference between the two different genotype classes. Okay, so this is a GWAS. We want to figure out what's the gene, what's the mechanism. I don't have that whole story, unfortunately, worked out, but there's one candidate gene that really stands out to us. That is a gene that actually people at Hopkins have studied for a long time, called polo-like kinase 4. PLK4 is one of the main regulators of centrosome duplication, the centrosome being the organelle that organizes the mitotic spindle. And it's really well studied in the context of cancer biology because it's known that overexpression of PLK4 causes the formation of more than 2 centrosomes. So rather than 2 centrosomes and bipolar cell division, if you overexpress PLK4, you can get the formation of, for example, 3 centrosomes. And then these multipolar cell divisions. In adult somatic cells, when you have 3 centrosomes or more centrosomes, the centrosomes actually tend to cluster. And so you still get a bipolar cell division. Actually, here's an example of that. Here you have 3 centrosomes, but the centrosomes cluster. So you still get a bipolar cell division, but you tend to get these abnormal chromosome attachments, like these Meratier-like attachments where it's attaching to both of the mitotic spindles and getting pulled in both directions. And so that can contribute to the formation of aneuploidy and ultimately to the formation of cancers. I think that in these early embryos, whatever the mechanism that causes centrosome clustering in adult cells is not happening in the embryos. And I suspect that when you have an abnormal number of centrosomes in early embryos, you often get actually tripolar cell divisions that you don't tend to see in these later differentiated cell types. And so here's some of our evidence of that. We can simulate what would happen if normal replicated genome segregates randomly on a tripolar spindle, right? So you have 46 chromosomes in a given cell. The genome replicates, and so now you have 92 chromosomes. And then if it divides on a tripolar spindle, each of the daughter cells should get about 30 to 31 chromosomes. And if we look in the actual data, what we see is a peak right at 30, 31. And then it looks like maybe a second peak around 20. I think maybe what's happening is these are cells that have undergone 2 consecutive rounds of tripolar division. So first one brings you down to 30. That replicates to 60. 60 divided by 3 is about 20. So it seems like this hit seems like it may be associated with increased rate of these multipolar cell divisions, potentially via centrosome amplification in the embryo. And actually, we have some little bit more direct evidence of that by pulling in time-lapse data from a subset of these embryos. We can identify which of these embryos underwent these unbalanced cleavages. So this is showing tripolar division in the first cell division, where the zygote, you can see it here, zygote directly divides into 3 cells rather than 2. But the same thing can happen on the 2nd division or on the 3rd division. And we see that the risk allele is associated with higher rates of these abnormal tripolar cell divisions. Okay. Okay, this is not the conclusion of the entire class, but just for the specific work from my lab, I showed you, well, this is even like background. Pregnancy loss is common in humans. It's estimated that fewer than half of conceptions survive to birth. And these abnormalities in chromosome number, which include aneuploidies or polyploidies, are thought to be the leading cause of pregnancy loss. Most of the meiotic aneuploidies are originating in egg formation in the maternal germline, and those increase in incidence with maternal age. Meanwhile, mitotic aneuploidies that are happening after fertilization are also very common, and we think that those are playing an outsized role in the very, very, very early pregnancy losses. Wouldn't even call them pregnancy losses, embryo arrest prior to blastocyst stage. Okay. Questions about that part before I move on? Yeah.
I was just curious, are there any other factors that are associated with meiotic aneuploidy, like smoking or and other environmental exposures or anything that has been determined?
Yeah, that's a good question. So in terms of environmental factors, I think we have really limited knowledge. The most, I guess, classic example is work from Pat Hunt's lab in mice, where they identified BPA as being associated with meiotic chromosome abnormalities in mice. And that's one of the reasons why BPA has been removed from all sorts of things. But actually her lab has recent work showing that the different plastic replacements for BPA do exactly the same thing, which is disturbing. But I actually think that, so my lab is trying to, right now, use those larger data sets to do more kind of quantitative genetics, like estimating the heritability of aneuploidy and recombination phenotypes. And consistent with kind of quantitative genetic theory, the heritability is low. So we're doing all this work to map out these genetic variants that are associated with the phenotype. But I actually think environment plays a much larger role in variability in these phenotypes. Age explains 20% of the variance, but there's still 80+% of the variance that's either driven by extremely rare variation. Actually, I think that's probably also playing a role, rare genetic variation, and environmental factors that we're yet to learn about. Good question. Common genetic variation in meiotic and mitotic machinery is associated with risk of aneuploidy. Probably also rare variation, just we don't have the methods or the statistical power to really be able to pull that out well. Okay. In second part of class, I want to talk about broaden this even beyond pre-implantation genetic testing, but talk about how are these tests actually being used in the clinic. I've focused the entire lectures up until now talking about how I'm repurposing those data to learn more basic things about aneuploidy and human reproduction, but how can this actually be useful clinically? So I guess just as some background, and this, what you're hearing today is my opinions. Some of the things I say may be controversial, and I'd like to hear your thoughts as well. But in the United States, there's very little regulation around prenatal and pre-implantation genetic testing. It's not under FDA jurisdiction. And so genetic tests in pre-implantation prenatal setting only have to be what's called CLIA certified, meaning that you show, I guess, like the precision of your test, that your test can kind of detect what you say it can detect, but the standard for like clinical efficacy is very low. You don't have to show that it's actually like beneficial to patients clinically. You just have to show that the test works technically. And so there's been a propagation of tons and tons of add-ons to IVF that are marketed to patients, which have, in some cases, no evidence of benefit, and in other cases, kind of controversial evidence. And so pre-implantation genetic testing, I think, falls into that second category, where it's been around for a long time, since the early 1990s, and now it's used in more than half of IVF cycles in the United States. But the evidence of clinical efficacy is still mixed. It wasn't until 2007, so about 15 years after PGTA was first rolled out, that the first randomized clinical trial was published in New England Journal of Medicine. And it made a huge controversial splash because it showed that at the time it was called pre-implantation genetic screening, did not increase, but instead reduced the rate of ongoing pregnancy and live birth after IVF for patients of advanced maternal age. And so that was concerning. The whole goal of doing this screening is to try to improve pregnancy outcomes, and probably, it depends. Actually, this is also a debate, like what outcome is the most relevant? What should we be measuring? But if you measured it in terms of like the overall probability of live birth after doing IVF, patients that didn't do this screening actually had higher rates of going home with a baby.
But of course, the question is, were the baby healthy, or they had trisomies or trisomies?
Yeah. So
that would be something at least. I mean, I know that nobody can tell you when you take home the baby, is this going to be a healthy baby? But you can tell like the big things, like.
Yeah. So the test, especially at that time, only was capable of detecting whole chromosome aneuploidies, and trisomy at live birth, I guess in the sense of like genetic disease, it's common, but in the kind of overall absolute percentages is very rare. And so I think in a clinical trial like this, you would almost never see like a trisomic live birth in either arm, like either the PGTA or non-PGTA arm of these clinical trials. But there are other outcomes that are definitely relevant, like, for example, miscarriage. How does it influence the rate of miscarriage, or how does it influence the number of transfers necessary? Patients undergo multiple cycles of IVF and potentially multiple embryo transfers. And so the alternative to doing pre-implantation genetic testing is just to sequentially transfer embryos with good morphology. So the reason for why this pre-implantation genetic screening, which has now been rebranded to pre-implantation genetic testing for aneuploidy, seemingly doesn't work as well as we might have hoped is an area of active debate. And I don't have all the answers, but one of the reasons, and certainly for pre-implantation genetic testing as it was done in the mid-2000s, where they were using cleavage stage embryos, one of the potential reasons that it didn't work as well as we might have hoped is this phenomenon of chromosomal mosaicism, right? You're taking a biopsy here, it's displaying it as like a blastocyst stage embryo, but at that time it was just taking a biopsy of one cell out of an embryo that was 8 cells, and we already know that, or we now know that mosaicism is very common, especially at the cleavage stage. And so your biopsy in that case may not be representative of the rest of the embryo. And so the whole logic of doing pre-implantation genetic testing starts to break down when you deal with that issue, right? So actually I had you guys read an article that was from 2015, which came out soon after. There was another publication in New England Journal of Medicine, this Wall Street Journal article is maybe more recent, but in 2015, there was a report in New England Journal of Medicine about the outcome of intentionally transferring a mosaic embryo. So prior to that time, if an embryo was determined as mosaic, it went into the abnormal bin and would not be transferred in IVF. There are a substantial number of patients that only have mosaic embryos available for transfer in a given IVF cycle. And so in relatively rare circumstances, a doctor may be willing to try transferring one of those embryos. And so that was done in this case report in 2015, and it led to a seemingly healthy birth of a baby after the transfer of a known mosaic embryo. And so that was this was one of the first big controversies. Here was a second big controversy, because for so long, patients were told they had no viable embryos available for transfer. Mosaic embryo would never be considered in IVF. But now it's shown that, it actually can lead to a healthy birth. And so this article that I had you read was talking about this difficult decision about whether or not to transfer one of those embryos. Again, in the United States, it's like the Wild West. Every clinic is treating these differently. There is no consensus in the field about what to do with mosaic embryos. And actually, even the companies that are doing the testing will decide to either report or not report that information in different ways. Sometimes they bin mosaic embryos as normal and just return a normal abnormal result to the doctor and the patient. Sometimes they actually report mosaicism and then leave it to the doctor to decide. This all leaves aside the question of, can you even accurately detect mosaicism, which I'll get to in a second. Yeah, Daniel.
Has there been any follow-up on those births to see if it's continued to be normal?
Yeah, so a little bit. I actually have a friend who has established a registry where fertility clinics can report the outcomes of mosaic embryo transfers, and those, I don't know how long they're followed up, in pediatric care or whatever, but at least the set of tests they do at birth, the babies seem to be healthy. And actually, okay, this is just from, sorry, my thing is covering up, just from 2021. There was the first randomized clinical trial of mosaic embryo transfers, tracking the outcomes of what happens when you transfer euploid testing embryos, versus embryos that look like maybe they have just like, 1 or 2 aneuploid cells within the biopsy versus embryos that have like 50% aneuploid cells within the biopsy. And measuring these different outcomes like pregnancy tests, pregnancy loss, miscarriage, live birth, mosaic embryos, regardless of kind of the class of mosaic embryos, had outcomes that were statistically equivalent to transferring normal embryos. So that was also shocking. Like, it seems like, at least in samples of this size, detecting mosaicism is effectively like equivalent to normal embryos, which raises the question, like, how should these be managed in the clinic? I'll turn that to you. What do you think? What would be your guidance? So we know that there are mosaic forms of trisomy, like, that are pathogenic. There are individuals with trisomy 21 of mosaic form that must have originated through these abnormal cell divisions. So it feels risky, but these data maybe suggest otherwise. Any thoughts about that? Even broader, what did you think about the article?
I think there is always a balance between what's good for at the population level may be different at the individual level. Because at population level, 0.9 or 1.1, whatever, doesn't make a difference. But if it's you, it makes a huge difference, right?
Yeah.
So
I think fertility clinics are also, tend to be very conservative. They're also worried about liability. Like, if they know that there's this abnormality that was observed on the clinical test, they may be very reticent to transfer that embryo, even if these data suggest that, there's no evidence that they would lead to aneuploid live birth. Yeah, Carter.
So I had 2 thoughts about that. I mean, the first one was, I remember in the article, they queried parents, and I think it was like 80% said they wanted, if they hadn't been told, or if they had been told, they wanted to be told. And so I think that, at least in the customer is always right mentality, that's right. I think the second thing is, I worked for like a year and a half for a little like male fertility company, like testing, they had some novel screening of sperm, age, whatever. Anyway, it was kind of hokey, but I did think it was pretty interesting fertility space where I think a lot of people just want to have as much information as possible, even if it doesn't even help you make decisions. And for a lot of people, like, it's very like mentally comforting to know, like, oh, I did every test I could have done, and I just know that I didn't leave any stone unturned. So if we still can't have a baby, then like it's out of our hands. And I think that's an interesting factor that plays in fertility that I don't know if is that present in a lot of other like medical fields.
Yeah, I totally agree with you. Like, and I don't, I'm a parent. I don't fault people for like wanting to do everything they can to ensure that, their kid is going to be healthy. But I also think that it's led to these incentives for companies to, potentially take advantage of that. And there's very little regulation around it. And so there's natural selection that happens in the industry space because of that. Yeah, Danny.
I also just find this fascinating how they all perform the same. It makes me wonder if, with the diagnostic test, if it's very useful at all, in the sense that all you're getting is the trophoectoderm, right? So you're just getting the outer layer. You said it becomes the placenta, right? So first off, if it only happened in there, it might not affect the kid later anyway. But also, who knows what the chance is that your euploid embryo, the inner cell mass is a mosaic, right? Because if you have the same, you'll have the same issue backwards.
That's right. Yeah. So I'll show you some data on that in a minute. But I would say that, okay, despite these complications, like if you could accurately identify meiotic origin aneuploidies, it should be relatively, logical that those embryos you would not want to transfer, because either they're going to lead to implantation failure, miscarriage, or cause disease in the baby, right? That's a big if, though. Like, can we actually identify? Can we, based on a biopsy, actually distinguish between meiotic aneuploidies and these mosaic aneuploidies? And I think that's actually kind of where the field's going, my sense of it right now. Like, there's increasing recognition that, like, okay, maybe mosaicism, maybe it's partially just like technical noise that's being called as mosaicism. Probably sometimes it's actually capturing like low level mosaic aneuploidy within the embryo, but the consequences for that for development are not very severe. But those should maybe just be treated as kind of like normal embryos. And meanwhile, we should try to focus on selecting against the meiotic origin aneuploidies. I'm going to skip that part. That's from my own lab, but just to say really briefly, we're trying to mathematically model this. So you're just biopsying like 5 cells, so you're getting an incomplete picture of the embryo, but you can try to extrapolate that to entire embryos, right? So given that you're just biopsying a few cells, well, assume that that's like a sample of the entire embryo, extrapolate to the entire embryo. We use an approach called approximate Bayesian computation that's based on simulations. But if you do that and you just make like some really simple assumptions, like for example, even mixing of cells across the entire embryo, pretty much regardless of of your assumptions about how aneuploid cells are mixed within a mosaic embryo, what we find is that to be able to explain the observed data are that like something like 18% of embryos show evidence of mosaicism within the biopsy. In order to explain that, you would have to assume that pretty much every embryo is either aneuploid or mosaic. At the blastocyst stage of development, when the embryo is like 250 cells, we think that every embryo has at least some aneuploid cells to be able to explain the observed data. And that, it turns out, is actually also robust, even if you assume that a lot of times you misclassify the biopsy. Even if you think it's like largely technical, if at least like, 10 or 20% of the time you're right, you still get this basic result that practically every embryo has some aneuploid cells. And actually this is not our work, but work from another lab, people are now using single cell sequencing on embryos to try to look at the chromosome complement of the individual cells. So here's a heat map of 3 different embryos. Each row here is a cell, and each column is a different chromosome. The number of copies of the chromosome is indicated by the color. And you can see evidence, for example, of meiotic aneuploidy. Here's an example of meiotic monosomy of chromosome 15 in this embryo. But you can also see evidence of these mosaic aneuploidies. Here's a monosomy of chromosome 18 that's present in about half of the cells. And so that probably originated during one of the first cell divisions of the embryo. But it's also very common to see these lower level aneuploidies, like a trisomy of chromosome 4 that's only affecting one of the sampled cells, in this case in the inner cell mass. So all of these methods, they have some, risk of false positives, false negatives, etc., that we have to worry about. But I think there is growing evidence that chromosome mosaicism is like a normal physiologic feature of early development. It's a little nuanced, right? Like severe mosaicism is the reason why embryos arrest prior to blastocyst stage, but conditional on surviving to the blastocyst stage, there's still like this typically a small fraction of abnormal cells within those embryos. So we were all probably mosaics. We still are, even in early development. And so here's what it looks like from the single cell sequencing data. And somebody mentioned, I think Daniel mentioned, like the possibility of aneuploidies being restricted like to the trophectoderm versus shared between the trophectoderm and the inner cell mass. And you see both, apparently, by doing the single cell sequencing, it looked like about half of the aneuploidies that were detected in the trophectoderm were also detected in the inner cell mass. And about half of them were solely restricted to the trophectoderm.
I
Okay.
guess this also goes for the villi testing. What did they call it? I forgot.
Embryonic villus testing.
Yes. Because I did it, but that would be the same problem.
Yeah, so by that stage in pregnancy, I think there's already been a lot of selection, but it's known that the placenta can tolerate chromosome abnormalities better than other cell types. And so there's recent work like doing single cell sequencing and like microdissections of placenta, and you see really frequent large, not just aneuploidies, but like large kind of segmental chromosome abnormalities. Okay, so I showed you that 2005 or 2007 clinical trial of pre-implantation genetic testing. Since that time, it's been rebranded to PGTA. It's been improved in some ways. Like now they don't do cleavage stage embryo testing, they do blastocyst stage embryo testing. They do sequencing rather than fluorescence in situ hybridization. So there are these kind of technical improvements, but the latest round of clinical trials was done in 2019, and it came to effectively the same results. Pre-implantation genetic testing had no improvement to ongoing pregnancy rates compared to not doing pre-implantation genetic testing. However, it's a relatively small sample, so it's still possible that maybe it's like reducing miscarriage rate a little bit. They don't see that in the data from this study, or reducing the number of necessary transfers to achieve pregnancy. I think there is kind of a consensus that it does reduce the number of transfers necessary to achieve a pregnancy.
Did they show data on that?
I don't know about in this paper, but I've seen, I think in the other, there were two clinical trials that were done at the same time. I think maybe in the other one, I think this one's called the STAR trial and the other one's called ESHRA trial or something. I think in the other one they show that. Is this field sketchy? Like they made a big deal about this result, P equals 0.03, which shows an improvement in the subset of patients from age 35 to 40. As a statistician, I'm like, you can't do that. You can't like post hoc like find a little like subgroup that has like a so we call this p hacking, but anyway, it's still, I think it's an open question of what outcomes does pre-implantation genetic testing actually improve? It's a screening test. It can't create healthy embryos. So kind of the best I think it could possibly do is to reduce the miscarriage rate, which this trial does not show, or reduce the number of necessary transfers in IVF. I told you it's used in like more than 50% of IVF cycles in the United States, but other countries have different regulatory landscapes. And in the UK, for example, there's much more strict regulation around prenatal testing by this Health, Fertilization, and Embryology Authority they use this like traffic light system. PGTA was given a red light because they said there's no evidence from randomized controlled trials that it's effective at improving chances of having a baby. But again, PGTA is just like one of the tests. Carter already mentioned like some tests you can do on sperm. There are tons of tests that are offered to patients in this space. Not all of them genetic tests, but many of them have these same types of issues. One test that is extremely common and is actually pretty mind-blowing that you may have heard of is called non-invasive prenatal testing, or NIPT. It's based on doing sequencing of DNA extracted from maternal blood draw at 10 to 12 weeks of pregnancy. And it turns out that at that stage of pregnancy, about 10% on average of the cell-free DNA in the maternal blood is actually of fetal origin. It comes from the placenta. And so you can identify aneuploidies in the fetus through the sequencing of the cell-free fetal DNA. By that stage of pregnancy, 10 to 12 weeks, there are only a few aneuploidies that survived that stage. Trisomy 13, 18, 21 are typically the trisomies that are looked for on that genetic test. And the test works very well. It's very, I think, compared to pre-implantation genetic testing, the evidence supporting NIPT is very strong. And for that reason, like, I think it's destined, right now, it's typically indicated for women older than 35 or with like history of infertility issues, certain fertility diagnoses. But I think eventually this will, if it's cheap enough and if access issues get dealt with, it will become kind of standard of care for all patients. There's still the possibility of false positives. Even though this test is very accurate, because aneuploidy is rare at that stage of pregnancy, more than half, I actually don't know the exact number. I won't put an exact number on this. A substantial number of positive test results turn out to be false positives. That's a general, kind of paradox from medical screening. You see the same thing in breast cancer screening with mammograms, or prostate cancer screening. If the incidence of the actual, condition is low, the actual disease is low in the patient population that you're screening, even if the test is very accurate, most of the positives will be false positives. And so you have that problem in NIPT. And for that reason, it's still only used as a screening test rather than a so-called diagnostic test. So it's kind of a first pass. And if an abnormality is identified, they still have to follow it up with amniocentesis or chorionic villus sampling, which is the invasive biopsy of placental tissue or amniotic fluid. Questions about NIPT? So actually the reasons for false positives, let me check the time. Reasons for false positives on NIPT are kind of interesting. I think they're not completely known. It's an area of research, but one of the reasons is abnormalities, somatic chromosome abnormalities of maternal origin. So again, like only a subset of this cell-free DNA is actually of fetal origin. And so what you're sequencing is a mixture of cell-free DNA from the mom and from the baby. And so one reason why you might see a chromosome abnormality in those sequencing results is like a tumor in the mother that she might not know about that's giving off this cell-free DNA and then is picked up in the NIPT data. That's been its own controversy about how do you deal with secondary findings from genetic tests. Do you tell the patient? Probably in that case, you would want to tell the patient, but the patient, at least at first, when it was not known that this was a possibility, there's a big question because the patient didn't sign up to learn that. And so do you return a result like that? Yeah, Zoe.
I just had a quick question. This might be a stupid question, but how do they parse out between maternal DNA and fetal DNA?
That's a good question. So I think in most cases, they don't really. They try to estimate the fetal fraction. So for that, it's a good question. I don't know the details, but for one, if it's a male fetus, you can use the ratio of Y chromosome reads to X chromosome reads or autosomal reads to try to figure that out.
Okay.
There's also a difference. There are a few differences. So cell-free DNA has different characteristics in terms of the fragment lengths. I think it tends to be shorter, and also the location relative to nucleosomes. Like nuclear DNA will have some pattern that depends on where it's bound to nucleosomes that's different than the cell-free DNA. And so those, I think, 3 characteristics can tell you information about the fetal fraction, and then you can use that information to kind of, you do that coverage plot like I showed you, but it kind of sets your expectations for where you should expect to see those deviations. And if the fetal fraction is too low, it kind of lowers your confidence in the calls altogether.
Okay.
Yeah, that's a good question. So far I've focused this entire thing on aneuploidy, but there are other, you can also do tests for other aspects of the embryo's genome, right? Not just de novo mutations, but you can also scan for inherited variation that is transmitted from the parents to the offspring. And that can be useful in the context of this test called pre-implantation genetic testing for monogenic disorders. It used to be called PGD, pre-implantation genetic diagnosis. Now, again, it's been rebranded to PGTM. But that's the idea that, for example, if the mother and the father are both known to be heterozygous carriers of a disease-causing mutation, which may have been identified through carrier screening, for example, which is common in certain populations like Ashkenazi Jewish populations or other populations where known to carry these founder mutations at high frequencies. They may undergo this carrier screening. You can do IVF, and then by doing effectively the same genetic test that's done for aneuploidy, but now looking to see which haplotype was transmitted to the embryo at this particular locus, you can figure out which embryo inherited the disease allele and which didn't, and then select the embryo that or prioritized the embryo that did not carry homozygous disease-causing allele. So that's pre-implantation genetic testing for monogenic disorders. It's been around for, I think, relatively long time now, and is kind of broadly, I'm sure there's still controversy about it, but I think broadly is sort of ethically accepted in the medical community. However, I had you read an article about how we're in this new world now where we can identify genetic variation across entire genomes. And there have been all of these association studies, genome-wide association studies, associating genetic variants with disease risk or other traits. You know, it doesn't necessarily have to be a disease. It can be height or IQ or skin color or whatever. Like, we have a pretty good understanding of the genetic basis of a lot of those traits. And so there's this, development in the field of what are called polygenic risk scores, which you may have covered in your other classes, rather than just looking at an individual variant and trying to predict risk of a trait. We're now in this GWAS world where any individual variant for complex disease typically only explains like a small fraction of the variance in the disease, right? And so knowing your genotype at like one of these sites isn't going to be helpful for an individual patient to really, unless you carry like a rare, like highly penetrant, large effect mutation, it's probably not going to be useful to know your genotype at like one of those sites. But you can add up kind of all of your risk across all these known associations and try to predict something like hypertension. And actually I think this paper is one example where they show that, okay, the polygenic risk for hypertension actually is like as good at predicting this as knowledge of like one large effect disease causing mutation. And so you can actually, maybe target interventions for patients, like start on this drug or, do this exercise regimen, whatever, based on this knowledge. Okay, that's in the context of like an adult population and a trait like hypertension. But you can imagine doing the same thing now. We can sequence the embryo's genomes or do array genotyping on the embryo's genomes and compute polygenic scores on embryos for a whole range of things. In the United States, there's no regulation around it. And so there are already companies that have sprung up and are doing this. I know some of the founders of these companies. And it's extremely controversial. One of those companies is called Orchid. Another one is called Genomic Prediction. And right now, at least they say, they're doing it for these, like, focusing on these disease traits like heart disease, breast cancer, whatever, and assigning these scores to embryos. This is also, complicated, obviously. There are many different traits you can try to predict. Which trait do you actually care about? What if like one trait's going in one direction, another trait's going in the other direction? Yeah, Carter.
So, like, personally, how much do you believe the statement that, like, they can say 27 out of 100 people with the same genetic risk as your lowest genetic risk embryo are diagnosed with heart disease? Like, how much do you buy that on a piece of paper?
I think it depends on the trait. Yeah, I have to read and think about that statement more carefully. But polygenic risk scores have been validated for some traits in like living populations. I think that's a different question than predicting something for the embryo, though. Like, by the time the embryo manifests this disease, it's going to be decades. The environment will have changed. The available medical interventions may have changed. So I think that is a big open question. I think it's going to depend on the genetic architecture of the trait and things like gene by environment interactions. I have a whole other set of issues with this. But obviously, like, this is not happening in a vacuum. Like, hopefully you've covered in some of your classes or, even history class, like, long history of eugenics and misuse of genetic testing. So I guess regardless of whether this works, there's an ethical debate about like whether this is the right thing to do. What is the line between something that we would call a disease and may be appropriate to like look at in like PGTM versus like what is just a trait? And that
we'll have a lecture from Deborah, actually two.
Oh, okay.
Yes.
Great. It's complicated. Like, I think it could be viewed as paternalistic to tell patients, like, no, you can't know this information about your embryo, right? Like, at what level is it an individual's decision to make reproductive decisions about their potential kids? But so here, I certainly think it's being over marketed right now, and it's very dubious. Like these, how do you combine the information from multiple traits? Even like technically, like, okay, one trait goes in one direction, one trait, the risk is in the other direction. They're creating some type of like integrated embryo health score and then coming up with a ranking of the embryos, but maybe you care about one trait more than another trait. And so there have been some papers in literature that try to like address both technical questions about polygenic scores and sort of these ethical questions. And here's just one from Peter Vischer and others. Peter Vischer is like one of the most prominent quantitative geneticists from Australia. Some of their points were that polygenic scores, much of the benefit of doing polygenic scores in like an adult like living population comes from comparisons between unrelated individuals. But here, because you're looking within a set of sibling individuals, you've reduced the variance immediately by 50%, right? So like you've kind of narrowed the possible value of polygenic scores substantially just by doing this comparison within a family. This is a broader challenge in the field of human genetics. Like, how do we convey uncertainty and like absolute risk versus relative risk? We know from like animal breeding and artificial selection experiments that the phenomenon of pleiotropy is widespread across the genome. That's where a given genetic variant doesn't just influence one trait, but it's simultaneously like correlated with many other phenotypes. And so when you select on one phenotype, you are, with or without knowing it, simultaneously selecting on a whole suite of other phenotypes. And so I think in human genetics, we still don't have a great grasp on kind of how is pleiotropy distributed across traits and how is that going to play out when you do this selection on embryos. And then the last point is really a broader, again, issue with the medical system. Like, the first adopters are going to be the people who can pay for it. And so does this have the potential to kind of exacerbate disparities across different populations? So I guess in the last couple of minutes, like, any other thoughts on polygenic scores. One interesting observation from the field is that I think everything is moving, like pre-implantation genetic testing for aneuploidy and pre-implantation genetic testing for monogenic disorders used to be separate like assays, different technologies used for one versus the other. I think in the future, everything will move toward genome sequencing, and it will probably be possible just from the cell-free DNA tests to get entire genome sequences out at that early stage. And then you'll be predicting a bunch of things about a pregnancy, or in the context of IVF, it'll also be genome sequencing on embryo biopsies and prioritizing embryos. Where do you see this going? Is it a good thing? How should we use those data? It's going to be a big diagnostic dilemma, I think. We'll be able to see everything, and what do you do with that information?
I have a question for the class, which is more logistics. Is it okay to have a class on Monday, which is technically a holiday? We scheduled it, and the professor asked if we're still doing it. I will say yes, but if someone does not want to do it, please speak now or forever hold your peace. All right. I see no hands up. Okay, we're going to have it. I'll tell the professor to show up. Thank you.
I think as answer to both, first to Elena, we have a class before this one that should still happen on Monday, and so we'll still be at school either way.
Good.
But in answer to all the PRS stuff, I don't know where it's going. I hope that more information will be useful, but at the same time, when you get so much information and you have to process the data, it just takes time and resources that we don't really have computer-wise. One of the things that I found fascinating when I read through the article about this one that you sent out was they had marked, like, I think it was diabetes, I don't remember if it was type 1 or type 2, where the average is like a 0.1% risk, and this one was at high risk because it was at 0.4%.
Right.
And I'm like, sure, that might be all that a PRS can tell you, but at the same time, it's like, that doesn't seem very informative to me, at least, on whether it will be useful information clinically.
Yeah, so that's the issue of like relative versus absolute risk, like 400% risk, but like in absolute terms, still low. Yeah, Carter.
I think that, I agree with Daniel. I think we don't yet have the resources for these to be that helpful, and it's easy to be sort of hand wavy and say like, oh, well, it's just not that informative, or they're just not that powerful. But I think eventually when they are, I think it'll be like more of a philosophical problem of like, what is a good life? And like, is the purpose of life to be as free from like disease and suffering as possible? Like, do you never want to face, or your children ever to face hardship? Is that like a good life or is not? And I think that we'll have to like grapple with those as a society, whether or not we want to spend resources and time like eliminating any sort of like disparity or like suffering in human health.
Yeah. Great. Okay, I think I'll just wrap up with a couple of broad conclusions, but that debate is fascinating to me, and I'm sure you'll encounter it, later when you talk to Deborah as well. So these genetic assays of embryo biopsies or amniotic fluid or placenta or cell-free DNA, they're used for testing for different types of abnormalities. Right now, there's a big focus on whole chromosome aneuploidies, but you can also detect smaller types of variation. And eventually, I think we're moving to whole genome sequencing. We're going to be able to identify even de novo lethal point mutations. And it's going to be a big challenge for our field in the next couple decades, like figuring out which mutations like are actually so harmful that you would want to deselect those embryos for transfer. All those tests have their own advantages and limitations. Like, for example, the issue of biopsying, you're not getting a full picture. Genomic platforms have their own sensitivities and specificities for detecting aneuploidies versus other classes of mutations. But a lot of this is converging on whole genome sequencing, and even that, there are different flavors of it, but it's getting more and more and more accurate. We're getting more and more and more information. Usually more information is good, but there's a lot of uncertainty about interpretation of some of these results. And so there's going to be this big diagnostic challenge, and how do we integrate, for example, polygenic scores into this? Cool. Well, it was good to talk to you. Thanks for the engagement. I'm sorry I can't join on Friday, but I hope you enjoy those papers, which the first one is about, it's from Decode Genetics, doing sequencing of trios and mapping out recombination, which the paper doesn't address it, but we now know recombination is closely related to the genetic basis of aneuploidy. And then the second one is from Magdalena Zernicka-Goetz's lab, where they established these mouse models of chromosomal mosaicism and are looking at selection that's going on within embryos and the ability of, like, you know, a clone of normal cells to rescue embryos. Cool. Thanks all.
Thanks to you. It was great. Thank you.
Thank you
Thank
for the
you.
presentation and lecture.
Thank
It was
you.
great.
Take care.
Hey Siri, call Dad on speaker. Hey Siri, call Dad on a speaker. Hey Siri. Hey Siri. Ooh.